friends, today we are going to study about protein tertiary structure, under which we'll study about the globular proteins, taking the examples of myoglobin, cytochrome C, lysosome and ribonuclease. So first let's start with myoglobin. What is myoglobin? As you all know, myoglobin is a protein that helps to store and transport oxygen in muscle tissues. So myoglobin, it is a small protein that helps to bind oxygen, and these are found in muscles. Because myoglobin help to store and help in diffusion of oxygen in muscle tissues, therefore this particular protein is found in abundance in the muscles of diving mammals. Why so? Because they remain submerged, and because of which they need to use their muscles a lot. Therefore, in diving animals, the presence of myoglobin is much more compared to the terrestrial animals. Now this myoglobin is a simple protein which is of only single polypeptide constituting a total of 153 amino acid residues, and it also contains an iron protoporphyrin, or which we know as heme group. So this how the myoglobin looks like. It contains 8 such alpha helix which are connected by bends or beta turns, and the heme group, this red one, is present in a pocket surrounded by these alpha helix. So myoglobin backbone is made up of alpha helix, which is interrupted by bends, and some of which are beta turns. The longest alpha helix constitute of 23 amino acid residues, and the shortest alpha helix constitute of 7 amino acid residues. Now these helices are right-handed. 70% of these helices are right-handed. The amino acids in these helices are present in such a way that they help to stabilize the myoglobin structure via hydrophobic interaction. Now what does it mean? That means in all these helices, the amino acids which are hydrophobic in nature, these amino acids are present in the interior of the myoglobin so that they can interact and form a hydrophobic bond, and hence can able to maintain the stability and the structure of myoglobin. So that means these hydrophobic amino acids are present in the interior of myoglobin, and the polar amino acids will be located outside the myoglobin structure. Now this molecule of myoglobin, it is so compact that it can hold only 4 molecules of water. So such a dense hydrophobic core is mainly stabilized by hydrophobic interactions and van der Waals forces. And this type of core is typical for all the globular proteins. Now, in myoglobin, 4 proline residues are found. Among these 4 proline residues, 3 of them are found in bends. The fourth proline, it is found in the alpha helix that helps to generate kinks. Kinks means bends, that helps to generate bends that are necessary for tight packing. And there are amino acids like serine, threonine and asparagine which are found in bends because they are very bulky. Their side chain is very heavy. So they cannot present in the alpha helix because it will disrupt the myoglobin structure. So these are mainly found in the bends. Now this is the structure of heme group in myoglobin. Now in this, heme group means it contains iron. Now 2 of its coordinates is perpendicular to the planar. This one and this coordinate. These 2 coordinates are perpendicular to the planar structure. Among that, one coordinate of the iron is bonded with the side chain of histidine, whereas the other coordinate bind with the oxygen molecule. Now, this heme group is present inside the pocket of the myoglobin. And what is the importance of its being in the pocket? It is remain separated from the surrounding oxygenated environment. So the cells are oxygenated. So this heme group is buried inside the myoglobin so that they can remain separated from the surrounding oxygenated environment. What happens if it not remain separated from the surrounding oxygenated environment? This Fe is present in Fe2 plus form. Because of oxygenated environment, it can get converted into Fe3 plus form. Now this Fe3 plus form cannot bind oxygen. the heme has to be in Fe2 plus form to bind with oxygen. So to protect it from oxygenated environment so that it can remain in the Fe2 plus form to bind with the oxygen, the heme group is present in the cavity or remain buried in the myoglobin structure so that it can remain separated from the surrounding oxygenated environment. Now, apart from myoglobin, cytochrome C, it is also a globular protein which is a component of respiratory chain of mitochondria. It also contains heme group which is covalently attached with this protein. The single polypeptide chain of about 100 residues with a single heme group. Now here 40% of the structure is in alpha helical form and the rest are beta turns. Similar with ribonuclease, these are secreted by pancreas into small intestine and helps to digest ribonucleic acid or to break and hydrolyze ribonucleic acid. It consists of 124 amino acid residues having the combination of alpha helical and beta conformations. And it consists of 4 disulfide bond. Another enzyme is lysozyme, which is found in egg white and human tears. It helps to cleave the polysaccharide in the protective cell wall of bacteria. So that means it helps to prevent us from bacterial infection by digesting their cell wall. It constitutes of 129 amino acid residues. Like cytochrome C, it is also 40% of alpha helical structure and rest are beta sheets. Like ribonuclease, it also consists of 4 disulfide bond. And these alpha helical structures are found in the active site of the protein. So these in short are about various globular proteins. So now one question from my end for you all. You have to answer. Among the listed interactions, which of the interaction will contribute more for the stability of the core of a globular protein? Is it a hydrophobic interaction, van der Waal interaction, hydrogen bond, or electrostatic interactions? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.